Hey team, Dr. Jack Audy here, and in this series of videos I'm going to be taking you through inflammation and how inflammation contributes to disease as well as aging. And there's a really interesting intertwining of inflammation and diseases and aging. But before I do that, I just want to start with a video which summarizes inflammation in 5 minutes. Just sort of a bit of a refresher of what's happening in the inflammatory process, and then we're going to break down each step in detail in subsequent videos. So let's jump into it. First up, this beautiful image here was taken by yours truly, and this is the brain of a mouse model of Alzheimer's disease, which is a form of dementia. And in the red, we can see the immune cells of the brain called microglia, and they're actually undergoing chronic inflammation as they surround this disease-causing protein aggregate there. And so this is sort of inflammation happening live, which is a super cool video. And I'm going to go into why would there be chronic inflammation in a later video. But I just thought I'd start with this beautiful image here. Now let's jump into what is inflammation, how does it happen, and let's try cover it in maybe 5 minutes. We'll see if I can stick to that time limit. So the key things to inflammation really are blood vessels over here and immune cells, right? And here we have a resident immune cell. That's an immune cell that sits in your tissue rather than circulating around your blood. And in this case, it's probably a macrophage, a big eater, a macrophage just sitting in the tissue. And what it's doing is it's surveying the extracellular environment for any perturbations, any sort of anything going wrong in that extracellular environment. So what could go wrong? Well, a simple thing is tissue damage. So here we've spiked the tissue with a needle, but this could be rolling your ankle or anything like that. And what happens is we get damage to the ordinary cells of the tissue. Now, what do we call those cells? We call them parenchymal cells. That means tissue cells, parenchymal cells. So that's kind of a universal term. You can use that anywhere in the body for tissue cells. So the parenchymal cells are now damaged, and you can see that there's now punctured membranes, for example. What does that cause? Well, that causes compounds inside the cell to now come outside the cell, right? So the extracellular environment is normally very different to the intracellular environment. So if you damage a cell, the contents of the intracellular environment are going to leak out into the extracellular environment. And that can be detected by our resident immune cell as it surveys the extracellular environment. Now, those compounds could be detected on extracellular receptor, so that's a membrane receptor that's on the surface of the macrophage, so it's poking out and it detects it directly, or it could be in the cytosol. There are actually a number of receptors in the cytosol that detect those perturbations. And so it has to get from outside to inside, and we'll go through that in a later video. But a really good example of this is ATP. So ATP, obviously, it's the energy molecule of the cell. It shouldn't be outside the cell. It should only be inside the cell. So when cells pop because of tissue damage or a metal spike, that is when ATP will leak out, and that can be detected by receptors on the macrophage. Now, these molecules that leak out that our immune system has learned to detect are called DAMPs. Damage-associated molecular patterns, right? So these are molecular patterns that are leaked out during damage. So damage-associated molecular patterns, and that's what the immune cell is detecting. Now, ATP is a DAMP, but there's loads of other different kinds of DAMPs out there. Now, probably what people more associate with inflammation and immune responses is infection, right? So there could have been bacteria on that spike, or there could have been viruses or fungus. There could have been pathogens in that. Now, pathogens are in many respects easier to detect because pathogens are coated in a surface of unique molecules that aren't normally in our body. The components of a bacteria aren't normally in our body, so when they are, our immune system has learned to detect them. So bacteria and fungus and even viruses contain molecular patterns on their surface, or they secrete molecular patterns that we can recognize as pathogenic. So they can, again, bind to different receptors. Sometimes there's even overlap. So DAMPs bind to the same receptor as a PAMP, but they bind to receptors on the surface of the macrophage or inside the macrophage or resident immune cell. And that tells that immune cell that there is now a pathogen somewhere in the extracellular space. So we have now either sensed tissue damage or bacteria. That's the first thing. These immune cells are constantly sniffing the extracellular environment, and it's honestly not very different from smell, because smell is just a receptor-based recognition of things, and this is exactly the same. So the resident immune cell has essentially smelled either tissue damage or infection, or both, because when there's an infection, there's often tissue damage. So now, once those receptors have been activated, they will now begin a signaling process. So they will signal to initiate an inflammatory response. Now, there's loads of signaling molecules, but two really good examples are prostaglandins and cytokines. And I'll dive into those a little bit later. And so the prostaglandins and cytokines signal for the inflammatory process to begin. So one of the first things that happens with these cytokines and prostaglandins, histamine is another one, we end up with vasodilation, so the blood vessels swell up and they become permeable. Why would you do that? Why would you swell up as a blood vessel and become permeable? There's lots of reasons. One, we need more blood supply. We need, because this site is about to become very metabolically active, because it's about to have a whole bunch more immune cells and everything, we need that greater blood supply. Two, there are proteins in the fluid of the blood that are antimicrobial, right? There are complement proteins, but also there are antibodies in the fluid component of the blood. So if we become leaky and allow the fluid component of the blood to leak into the extracellular environment, we're allowing those antimicrobial compounds to leak into the extracellular space to begin to fight those pathogens, right? So that's why there are gaps here. So the blood vessel swells up, becomes bigger, and it becomes leaky because we need more blood to get there. We need the fluid. We want as much fluid of the blood to pump into the extracellular space to begin to fight those pathogens with those proteins that are almost magical in their ability to fight pathogens. But another reason is we also want more immune cells to get to that site, and immune cells get there via the blood. So if we can get more blood there, we can get more immune cells there in a shorter amount of time. Now, this process causes swelling, which we all know happens with inflammation, right? When you roll your ankle, it swells up. I think I'm going to go over 5 minutes, but I'm trying to explain inflammation. Anyway, so next thing is cell recruitment, and they are responding to those signaling molecules, okay? So they're being released from the bone marrow, for example, into the bloodstream, or they stick to the blood vessel wall, like this neutrophil here, and they squeeze themselves into the site. What immune cells should we expect to be there? Neutrophils for sure. They are like the quintessential inflammatory response cell. Neutrophils are a phagocyte, so they will eat stuff there, but actually one of the main things they do is degranulate, and those granules contain really antimicrobial compounds, and so they spray the area with antimicrobial compounds. And they actually induce the production of household bleach, something chemically identical to household bleach, to sterilize the area. So they spray antimicrobial compounds around. Now, a monocyte is another circulating white blood cell, and they come there and they kind of take over managing the immune response, okay? So they can actually turn into macrophages if they're there for long enough. They also take over the cytokine secretion, and they take over the phagocytosis. And this is because the macrophages can sometimes die during the process. They're there, surrounded by bacteria, they might die, so we need a cell to come over and replace the role of the macrophage, and that's often what the monocyte is doing, really crudely. Now, some of these processes you could see are potentially damaging. So the macrophage will start eating the damaged cells as well as eating the pathogens, and the neutrophils are spraying cytotoxic compounds like bleach to kill all the bacteria. But if that's ongoing, you can start to see how inflammation could contribute to disease. The macrophages could start eating healthy cells, or the neutrophils could continually spray their cytotoxic compounds around the place, and that kills even more of your host cells, of your cells. And so you can start to get collateral damage going on. So that's why inflammation can be a damaging process as well as a beneficial process. But we need to get back to normal. So the final stage of inflammation is resolution. We need to turn off that inflammatory process. We need to clean up the parenchymal cells. Then we need to replace the parenchymal cells. and the extracellular matrix, which the parenchymal cells are sitting in, to get back to normal. Okay, so that's the final stage of inflammation, is resolution to get back to normal. Now, I'm not sure if that was 5 minutes, but up in the next video, we're going to cover that sensing step in detail. So we're going to go through each of those steps, sort of sensing, response, and resolution, sensing, signaling, response, and resolution. We're going to cover each of those steps in subsequent videos. And so up next is, how do we sense those pathogens?